Good. So then what do you get in glycolysis from 1 glucose? 2 pyruvate. What else? 2 waters. What else? 2 ATP. Good. What else? 2 NADH. And those NADH are in good. Okay, good. What comes after that? Glycolysis is first, then what? Aerobic oxidation. Good. And what do you get in aerobic oxidation from 2 pyruvate? 2 acetyl-CoA. Good. What else? 2 carbon dioxide. Good. What else? Good. Where are they at? Good. Good. Look how well you know that. All right. Good. What's next? Citric acid cycle. Good. And how many times through are we going to go if we have 2 acetyl-CoA? Okay. So 2 rounds. So in 2 rounds, what will we get? 6 NADH. Good. And they're in the good. What else? 4 CO2. Good. What else? 2 Good. One more thing. 2 FADH2. 2 FADH2. All right. Good. So that's hopefully what you have for number 4, right? I really want you to be thinking of breaking it into steps, okay? So that brings us to what is the net reaction for the metabolism of 1 molecule of glucose 6-phosphate? So this time we're not starting with glucose, we're starting with glucose 6-phosphate, okay? And so you need a glycolysis picture. Find your glycolysis picture. And find glucose 6-phosphate. And if you if just one person in your group has a glycolysis picture, that's fine. Just look at it together. But you need a glycolysis picture. And so find glucose 6-phosphate in there. Where's the glucose 6-phosphate? Okay. So what will you get? So first of all, how many carbons in glucose 6-phosphate? Still 6. So I'm still going to get 2 pyruvate. I'm still going to get 2 waters. Are you still going to get 2 ATP? You're going to have, is it going to be 1 ATP, 2 ATP, 3 ATP? How many am I going to get all together? 3. 3. Because you're using only 1 this time, right? You're skipping that very first step. So you're only using 1, but you're making 4. So net, this is 3 ATP. And that's really the only thing that's different. Okay, everything else about this is the same. What's more energy efficient? Is it more efficient to use glucose as a source of energy or glucose 6-phosphate? Glucose 6-phosphate, because you'll get an extra ATP. Okay, everything else is the same. Okay? And then that also means, you could turn those into total ATP, right? We practice that in class. So this is something like what you're going to do in your homework, is you're going to start with something different. So I want you to start with glucose. I want you to start with glucose 6-phosphate because sometimes you use glucose 6-phosphate, like in glycogen. And then we'll start with something else weird, like pyruvate or something else, and then figure out what you get in each pathway. Okay, how about number 6? Cheyenne, what do you have for number 6? What are the major inhibitors of the citric acid cycle? Good, ATP and NADH. And when I say inhibit, what does that mean? Does it mean to turn the pathway on or off? Off. Okay, good. And then what happens to the citric acid cycle if NADH and FADH2 are not reoxidized? That's for you, Courtney.
It stops working because there's not enough NAD plus FAD.
Totally. Yeah, so the Krebs cycle will totally be off. And where does this happen? That NADH and FADH2 get oxidized. What pathway oxidizes the NADH and the FADH2? The electron transport chain. Good. Okay. So that brings us to page 151. And so when it says, what is oxidative phosphorylation? And so this one's for you, Chris. What do you have for that?
So I have oxidation as a result of oxidation of NADH and FADH2.
Okay, good. So if I asked you who's oxidized in oxidative phosphorylation, what would you guys say? NADH and FADH2. And if I asked you who's phosphorylated, what would you say? ADP, right? ADP gets a phosphate group added to it. Okay, So number 2, how are the processes, this actually was a test question, that's why it's in here. How are the processes of citric acid cycle and the electron transport chain related? So what do you have, Amy? Okay, so the Krebs cycle supplies the NADH and FADH2 to the electron transport chain. That's part of it. What does the electron transport chain do for the Krebs cycle? Yeah, it oxidizes them. So the electron transport chain supplies the NAD and the FAD that's needed in the Krebs cycle. Okay, so they're synergistic. All right, so Yeltsin, number 3, why is the electron transport chain needed for ATP synthesis? Okay, so you really just need proton gradient, right? You need a proton gradient in the electron transport chain in order to make ATP, and that is what you get in the electron transport chain. Okay, how many ATP per NADH for a prokaryotic cell, Gabby? 3, and how about for FADH2? Good. And in a eukaryotic cell, the NADH and the mitochondria count for, and in the cytosol, good, and FADH2, good. Okay, good. So number 5, this is for you, James. What's the purpose of the glycerol-3 phosphate shuttle? Okay, so does anyone have ideas on the glycerol-3 phosphate shuttle? Like, what is the purpose of that glycerol-3 phosphate shuttle?
Give electrons to the FAD to turn into FADH to form DHAP.
Okay, sort of, not exactly.
Something across the outer
Definitely.
membrane.
It's to transfer molecules too big to pass through the cytosol to the mitochondria.
Okay, so let me just say this. That shuttle system, what's the issue? Like, what is the issue that you have in a eukaryotic cell, Leah?
So that you can make use of the NADH in the cytosol.
Good. That NADH in the cytosol can't cross into the mitochondria. And so this shuttle system is utilizing its hydrogen, right? That's really all that happens. So that hydrogen passes to DHAP. It becomes glycerol-3 phosphate. That crosses into the mitochondria. And then who is that hydrogen passed to from glycerol-3 phosphate? Who accepts it in the mitochondria? No, it's not DHAP. So it's passing into the mitochondria as glycerol-3 phosphate. Who's the acceptor? FAD, right? That's really important. So you're making it, it's like NADH up here in the cytosol, and it passes through a series of acceptors, and you're using it as FADH2 in the mitochondria, right? And that's why this NADH in the cytosol, it counts for 2 ATP, because we're using it as an FADH2. Does that make sense? So this NADH in the cytosol doesn't ever cross into the membrane. No, it doesn't. The only thing that is passed is its hydrogen, right? Its hydrogen is going to be carried by that glycerol-3 phosphate molecule. Okay, well, DHAP accepts the hydrogen, it becomes glycerol-3 phosphate, then it passes into the mitochondria. Okay, that brings us to number 6. So this is for you, Kat. How come the NADH in the cytosol is equivalent to 2 ATP, but in the mitochondria, it's equivalent to 3 ATP? Okay, well, we kind of just talked about it. This is the NADH in the cytosol, and we count it as 2 ATP. How are you using it in the mitochondria? What does it say right there? Yeah, and how many ATP are the FADH2? How much do they count for? They count for 2 ATP. So the point is, you're using it as NADH. It's made as NADH in the cytosol, but it's used as FADH2 in the mitochondria. Okay, how come, this is number 7, so this one's for you, Leah. How come the ATP produced from NADH, you get more ATP from NADH than FADH2? This is number 7.
Because it passes through the compounds to the NADH.
That's right, so it contributes more to that proton gradient. Okay, So then this brings us to number 8.
What are we supposed to
We're at
Rosie?
like
We're at
25.
milliliters here on
Yeah.
Okay, then you just need to flip it up and collect it.
And then
So-
switch down.
Yeah,
Okay.
totally. So let's talk about number 8 for a second. Really, this is what I hope to see in your number 8. I hope that you wrote out what you make in glycolysis, what you make in aerobic oxidation, and what you made in the citric acid cycle, and then you turn it into ATP. So if we were gonna do that, this NADH in the cytosol counts for how many ATP?
the solution to the total volume.
Well, each one is times 2 ATP, right? So this is 4 ATP plus 2 here. So that makes 6. Okay? What about this NADH in the mitochondria? How many ATP does that count for? 3. Good. And so how many ATP then from this step? 6. Okay. And then this NADH in the mitochondria is still 3 ATP. What about GTP? It just counts as ATP. And then what about the FADH2? How many ATP does that count for? 2. Okay. So this would be like 4. So 4, 5, 6 plus the 18, 24. And so how many ATP altogether? 36. Okay? And so that would be, if we're talking about a eukaryotic cell, that's what it would be. How would it be different if it were a prokaryotic cell? 2 more ATP. How come?
Because there's no difference between the cytosol and the mito.
That's right. So it's really this step right here in a prokaryotic cell, like that would count for 3 ATP. And so that would make this 8. And then altogether you would have 38. Right? So we don't have to worry about where the NADH is in the prokaryotic cell. Yeah.
So if you're asking us a question about like where, or how many ATPs or something, are you wanting us to say that 36 ATP is coming from the Krebs cycle?
It's not really. I would say 36 ATP is coming from a single glucose molecule. So from a combination of glycolysis, aerobic oxidation, Krebs cycle, and really the electron transport chain. But the important key thing is that we're assuming it's aerobic. Do you know what I mean? If it were anaerobic, then all of this would be off except for glycolysis. Do you know what I mean? You would have glycolysis and then what other pathway? Lactate fermentation, right? It would be different. So you wouldn't really make any ATP except for right up here. And even this, could I turn that into ATP if it were anaerobic? No, because this happens in the electron transport chain and that requires oxygen. Do you know what I mean? So if we were counting ATP for an anaerobic situation, it would literally just be those 2 ATP right there. Do you know what I mean? Like that's it. You can't turn the NADHs and the FADH2s into ATP unless it's aerobic. Does that make sense? That's a good question. Okay, so this brings us to number 9, and it says calculate the ATP produced from 1 molecule of pyruvate. So this is the kind of thing I'm going to ask you on an exam. So I'm going to ask you like, hey, we're going to start somewhere different. So if we're starting with pyruvate, where do you make pyruvate? Like where is that? At the very end of glycolysis. So do I have like a glycolysis pathway? Yes. No, I don't have that. I'm starting with pyruvate. So none of that is happening. I don't have any of that. And I'm not starting with 2 pyruvate. How many pyruvate am I starting with? 1. So there's really only, let me just read. There's really only 1 pyruvate. And I'm starting with aerobic oxidation. So you might think like this is made up, but this is a real thing. Like this would be an amino acid. Some amino acids directly feed in as pyruvate. So if on 1 pyruvate, how many acetyl-CoA would you get? 1 acetyl-CoA. What else would you get?
1 CO2.
Good. What else? Good. And that NADH is in the mitochondria and will count for how many ATP? 3 times 3. So this step would just be 3 ATP. Okay? And if I have 1 acetyl-CoA, then how many times through the Krebs cycle? Good. So 1 round. What will I get in 1 round?
2 CO2s.
2 carbon dioxides. Good. 3 NADH. Those are in the mitochondria, so they count for 3. Good. 1 GTP. Good. FADH2 count for 2 ATP, right? So this would be like 9, 10, 11, 12. So how many ATP altogether? 15. Does that make sense? So this is a good test question, right? I'll just ask you some other molecule, and you want to be thinking about what pathways are they starting in. Okay, and you're going to practice this tonight in some of your homework too. Okay, this brings us to number 10, which is how many molecules of the following are produced during the metabolism of one glucose? And it could be produced at some point. It might be produced and then used, but at some point, how much do we make of these? So Hannah, did we do you? Yeah, I can't remember. Okay, how many acetyl-CoA? 2 acetyl-CoA, that's right. And then Sarah, how many carbon dioxides during the metabolism of glucose? Altogether. So we're on number 10, B. 6 carbon dioxides, that's right. So if you kind of went back to what you hopefully wrote in question A, you're literally just counting these up. I just kind of erased those. Yeah, Katria.
So do we still say 2 even though the acetyl-CoA is used up?
Yes. So at some point you make 2, even though it's made and then used. But at some point you're making 2.
Okay.
And then so that brings us to pyruvate. What do you have for that, Katria? 2 pyruvate. That's right. And then Zach, how many NADH?
10 total, 2 in the cytosol, and 8.
Good. All right, good. All right, and then this brings us to number 11. So Amy, this is for you. What are the 2 mobile electron carriers in the electron transport chain?
The CoQ between 1 and 3 complex, and the cyto-C between 3 and 4 complex.
Okay, so those are cytochrome or coenzyme Q. That's what they stand for. So coenzyme Q, it moves between 1 and 3 and 2 and 3. And then the other one was cytochrome C. So it looks like that, cytochrome C, and that does move between 3 and 4. Okay, and then final products of the electron transport chain. Rosie, what do you have for that?
NAD plus, FAD plus, water, and ATP.
Good, not ATP. What do we make in the electron transport chain? So NAD plus, FAD, water, and what's the other thing? Not ATP.
Protein gradient.
A proton gradient, right? You need that proton gradient to make ATP. Okay, and then what is the last electron acceptor in the electron transport chain, Jay?
Oxygen.
Good, which becomes water. Yeah. Okay, good. So then I just want to make sure that we go through it because there is a lot of good test questions. You know, like a lot of what I'm going to have on your exam is showing up in your homework. And so it's important that we make sure we talk about it. Amy. So number 12, the final products of the electron transport chain, NAD+, FAD, water, and a proton gradient. And then number 13, the last electron acceptor is oxygen. Okay. All right, so then what I'd like you to do is set aside your notes. We're gonna take our quiz. So kind of clear your desk, put it off to the side. And we're gonna take this quiz, it's kind of a special quiz. We're gonna take it together. Well, you can take it with your partner, is basically what I mean. And so you're gonna agree on the answers, and I'm gonna give you each a copy, but really I only want one copy between you and your partner. Does that make sense? And you can work with, another group if you like. I just want you to be periodically keeping an eye on your samples, right? So if you're grabbing, you get to the 15 mils, collect your samples.
Renamed. What the fuck?
So,
So much water.
The quiz is up here, just come and grab it.
Grab us 3.
And then the last thing is, I'm going to give you like maybe 15, 20 minutes to take the quiz, all right? Because I need to talk about something about the experiment. So we want to be efficient as you work on this quiz. Okay, if you need to talk with another group, that's fine. But be efficient. maybe not your homework, but the rest of the experiment. So, listen, I'm gonna be passing back your amine homework. And I'm not sure, like, sometimes when you do homework, I realize you're tired and you just write down an answer. But what I wanna say is that there are test questions on here. And the way you're answering them, they're not gonna fly. You know what I mean? So if I ask you a question that says, how does cocaine affect neurotransmitters? That's a test question. But a lot of you are like, oh, we just get more neurotransmitters. And you need to work better on that. So I marked them. You should be getting them with your partner, and you have time. Like, talk about it with your partner because you need to be able to answer them properly. All right? And then there's a question, one of these. So it says something like N-N-dimethyl 2-butanamine. And so when you do these amines, you need to work backwards. So butanamine means how many carbons? 4. And what number carbon is the amine going to be on? 2. Oops, amine. Nitrogen. And then what's going to be attached to the nitrogen? 2 methyl groups. Okay, a lot of you are trying to put the amine on the end, but it's not telling you that. Do you know what I mean? So make sure that you look at that. So please, like, don't underestimate the amines. Do you know what I mean? They are on the next exam, and you want to make sure that you understand them well. Do you know what I mean? I'm a little concerned about that. Yeah. It wouldn't, because nitrogen only forms 3 bonds. Okay? There are some questions on here, like 2 reactions that you literally took a test on a week ago, and you all looked at them and said, I have never seen them before in my life, and invented your own reaction. Do you know what I mean? But what do you make when you have a carboxylic acid and an amine? An amide, yeah. And a lot of you are like, no, I've never seen that. Carrie never talked about it. But we just took a test on it. Okay, so make sure you please look at these. All right, please look at them. I'm almost tempted that we need to have a quiz on amines to make sure that I know you know them before the test. Do you know what I mean? Because I just want you to be as ready as you possibly can be. So let me talk about the last bit. So there's a few things for your little setup that we sort of need to talk about. So if you're still collecting samples and waiting on a last sample, there's a few things that you can be looking at. So look at your data page on page 87. And you see about halfway, well, towards the bottom of the page, it says, show the calculation for the theoretical yield of ethanol. Okay, that's a Chem 2A calculation. It's like the calculations that you did in the aspirin lab, where you started with your grams of acylic acid and you went to grams of aspirin. Okay, and so you need to do that same calculation here. If you look in your homework, it asks for the lab calculations, all right? And so that is part of it. The other part of it, as soon as you've started to get, well, actually, before that, you can also be practicing with a pipette. So you guys will need like a 10 ml pipette. And I want to remind you about pipetting for a second. So I would encourage you to practice pipetting. The bulbs are over here on the side. But I would encourage you and your partner to practice pipetting with a little beaker of water first so that you remember how to pipette. Like, you want to be accurate in this part of the experiment. So just to remind you, you take your pipette, and what do you have to do with the pipette before you can use it? Good. So I squeeze my bulb, I stick it on the end, and I suck up maybe 2 inches, okay? When you're conditioning, just a little tiny bit. So you guys are going to have to condition your pipette with your samples. But that ethanol that you use for conditioning uses up some of your product. So I want you to imagine that ethanol is liquid gold and don't waste it. Does that make sense? So you just use a little bit, run it along the inside of the pipette, and then the ethanol, you can just drain it into the sink, okay? So condition it at least twice, all right? And then if I were you, I would practice with water. Remember that when you use the pipette, you squeeze the bulb, stick it on, suck the liquid up. When it's in the fat part, it will go real slow. You'll suck the liquid up past the brown mark. Careful to never let go of the bowl, otherwise you'll suck liquid up into the bowl. So if it feels like it's not feeling enough on you, pull it off and re-squeeze it. Do you know what I mean? Although these blue bulbs are like ginormous. And once it fills the fat part, it's gonna try to shoot up into your bulb. So keep firm pressure. Fill past the brown mark. Be ready with your finger. Pull the bulb off. Put your finger on the end of the pipette. And then with a little pressure, not like your life depends on it, just a little bit, like loosen it. So you're just twisting. Oops. And as you twist, the volume will start to go down. If it goes down too far, like it just did for me, just re-squeeze it and set a little bit back up there. And then you twist, twist, twist until the meniscus is right at that brown mark. Then push like your life depends on it. Okay, and that is your 10 mils. So you should probably practice this with water, all right? And then once you practice and you and your partner feel confident, then for each of your samples, and so if you notice, it looks like on page 87 that there's 3 samples, but some of you are gonna have more than 3 samples, some of you might only have 1 sample. Do you know what I mean? It just depends. But once you have a sample and you have practiced with the pipette, then this is what you'll need to do. So you're gonna have your sample. The first thing you need to do is condition your pipette.